Tracy, my question for you is how are you and your team approaching safety monitoring in your programs? And do you see it shaping trial design or clinical adoption?
Yeah, absolutely. Safety is one of the strongest differentiators of NK cell therapies, especially compared to T cells. And it's something we take very seriously, especially considering my own child is a patient in this study design because NKs they naturally regulate themselves through a balance of activating and inhibitory receptors. The risk of severe toxicities like cytokine release syndrome or graft versus host disease is inherently lower. We just haven't seen it in any of our 350 humanized mouse models, in the 7 patients that we treated, and the 16 doses that we've given. We are actually seeing the opposite of toxicity. We're seeing patients actually feel a lot better. That being said, we're not leaving this to chance at all. We're embedding rigorous safety monitoring into our program from day one. That includes real-life cytokine profiling, immune function assays, and close observation of organ functioning during treatment. We've recently had one patient that was delayed by 4 months because we were waiting for those liver functions and those organ functions to go back to a safer state, because we just don't want to take any kinds of risks with these patients, especially in the early phases of building this NK cell platform and establishing safety. By building these strategies into trial design, we can both reassure regulators and accelerate adoption by clinicians who want safer, more protective immunotherapy options for their patients. And over time, we believe this strong safety profile will make NKs the preferred off-the-shelf therapy platform. When you have something that is so scalable and we believe is going to be quite cost-effective to manufacture as we continue that scaling process, we're in the optimization phase as we speak now at a lower scale, but we're hoping to very soon, not only are we optimizing to produce a lot more cells at a time, but we are scaling that up as we speak to prepare for our human trials in the United States. So we hope to begin early to middle next year once we get that IND approval. And looking at this palliative care model that we're using currently, we also hope to expand that globally and look at other trial sites and study sites that we can adopt, that where patients around the world can get access to this therapy more expeditiously versus having to wait for FDA approval in the United States, which we all know is a very, very slow process. So monitoring safety on the upfront for us is absolute paramount. We are extremely meticulous in evaluating medical records and making sure that the patient goes at a time that's going to be best for them. And we've got patients that are on combinational therapies right now. So there's also a lot of juggling around what are these other more toxic therapies doing to the patient's body prior to and after they're getting their natural killer cells, and really ensuring that that timing is perfect, as perfect as it can be for something that's so new and early phase. But we're very excited about how this is going to be looked at. We're excited about the strategies that we've deployed. We have our own set of diagnostic tests at UCLA that we're using that aren't available in the market today, where we're truly looking at that immune function on a cellular level. We're looking at those ratios of NK cells to T cells to gamma interferon release to cytokines to ensure that the patient not only is recovering from that broken state, but they're staying in a safety profile range that makes us all very, very comfortable in knowing that that patient is going to continue to improve and not get worse on our therapy.